The biggest obstacle for therapeutic in Alzheimer's has been the lack of understanding of the biology. So this is where Alzheon team really came in, and the four discoveries that were seminal actually for getting us what I consider now is the dawn of really us able, the new era that actually we would be able to not only treat the patient but hopefully actually also prevent the disease. Is the understanding of one, what is the toxin, what's causing the disease. Second, how you find the patients reliably, how you identify patients with truly Alzheimer's disease. Distinction between dementia and Alzheimer's has been very difficult. And the old diagnosis just based on clinical symptoms was about 30% wrong, which has been a big problem for drug development. The third has been how you can follow the progression of the disease and therapeutic efficacy with biomarkers. So we pioneered not only the imaging, which is the volumetrics, and we'll talk about a little bit more, because that's the best evidence for really changing the course of the disease if you can change the rate of the atrophy or stop the atrophy. But also the fluid biomarkers, which has been really the most important advance now. And then finally, the really uncovering the secrets of the what is the role of the ApoE4 genotype, right? So when I'm originally from Prague. I did computer science and medicine. I came here for grad school. In fact, I actually worked with Eric Kandel in his group here at Columbia. I think the Nobel Prize for Learning and Memory. But I wanted to work on learning and memory. So there was my advisor at the time found about the discovery of Alan Roses at Duke, who founded the role of ApoE4 genotype in Alzheimer's, which is now 25 years ago. And he really thought this would be a great project. So I reluctantly just started working on ApoE4 in Alzheimer's, and it has been really the thread, through all my work and career. And the understanding of the genotype, which drives almost 2/3 of the patient, have actually this genotype, has been essential for us for the first three discoveries that I just mentioned. For understanding the biology, understanding what is the toxin, how you treat it, how you intervene, how you follow these patients. And what really has been striking, because this was the first few papers we published at Alzheon, has been identifying how distinct these patients with either one, or actually more so with the two ApoE4 genotypes, the homozygotes are. We uncovered it, and Susan Abushakar, our chief medical officer, published 2 papers on this from a clinical data set based on the clinical efficacy actually of the predecessor molecule. And now one of the major breakthroughs has been the Fortea paper, Juan Fortea, who uncovered, who actually described from the sample of about 10,000 patients how distinct the ApoE4 homozygotes are. And basically what the point of his finding of this Nature Medicine paper is that if you have 2 copies of ApoE4, you will have the disease. You will get the disease. It's almost fully penetrant by age 65. Virtually everybody will have the brain pathology, and the vast majority of people will get in their lifetime the clinical symptom, the terminal disease. So these were really the pieces that had to come together, for this dawn of the new therapeutic era. So right now we have few symptomatic treatments. There are, as you probably know, there are a few antibodies that have been brought into the market. The limitations right now really for the current treatments is the meaningful efficacy. Can we actually improve the efficacy? Can we make these treatments safer? Especially avoiding the brain hemorrhage, what's called the ARIA. Can we improve the access? Because the problem is that if you have infusions of the antibodies, it's very difficult for the millions of patients to get the treatment. So our goal from the very beginning, our mission has been to develop oral treatment for Alzheimer's. That's been from the very, very start. So we are only focused on actually that. And I think the oral approach would dramatically improve the access to the treatment. The next one is so one of the discoveries has been the course of Alzheimer's disease. So what we know now, what we consider Alzheimer's, the clinical part of the disease, is the terminal disease. It's literally when the brain fails, and with all the destructions, killing the brain cells and destroying the synapses, the connection between the cells, and it cannot really recover the function. So brain cannot regenerate. There's what's called plasticity. So the parts of the brain that are still healthy can take over the parts that are damaged. And the point where you have clinical symptom is the utter failure, right? And again, there's a very substantial atrophy at that point. But what we know, and this is really where the opportunity lies, is that 20 years, maybe 30 years before that, the brain changes start to happen, and we can measure them. We have biomarkers that can directly follow the brain neurodegeneration, which is the most prominent, and it's called phospho-tau, p-tau, which is a marker that really shows that the brain cell has died. So we can find these patients a long time before they have the clinical symptoms, and hopefully, if you intervene in the pathogenic pathway, which we're doing, we can actually prevent them ever going to the terminal stage, the clinical stage. One of the other issues that we're trying to address, that's maybe the fifth point, is the patient journey, right? So the problem is that if we introduce a complicated treatment, like the infusions have been very difficult to administer because there are over 20 or 30, depends on which one is it, steps that the patients, the payers, have to do to really, before they can actually give the treatment. I mean, the advantage of the oral, and especially if we're focusing on these very high-risk patients, is just 2 steps. So it shortens and it shortens the patient journey and actually allows us to be much more effective. So, again, I said it 2 times, I said it the 3rd time, we're at the dawn of a new era. We understand the disease. That has been the biggest problem. People tried so many different approaches to treating Alzheimer's, and it all has been failing because we just did not understand the biology. We do understand the biology. We published a number of papers covering those, few things that I just mentioned, and now we will have effective treatment. And fingers crossed, our investigational drugs that is finishing now the phase 3 trial, hopefully can help patients and be used not just therapeutically, meaning in the terminal stage when people have symptoms where we now evaluate them, but also go earlier to the pre-symptomatic, where really you can make a difference for the millions of patients. What we now hearing that there's a cost of the infusions, but the cost of the infrastructure and kind of delivery of the care is almost 3 times as much, right? Think about it, right? So it's just really problematic for the healthcare system, and this is also the reason why the therapies that have been introduced have not been as widely used, because it's just so difficult for every step of the delivery system actually to bring it to patients. A huge advantage we had was this molecule, that it's like a probe into the biology, right? We really could explore what's happening in the brain in the patients, how to follow it, and it allowed us to understand the pathogenic pathway, which is the amyloid pathway, where you go from a healthy protein, which is the monomeric protein, into misfolded protein that forms prions and then start to aggregate. And this is the toxic part, the oligomers. And then, the brain protection against these oligomers is what's called the amyloid plaque, which has been the wrong target for many programs, and that's why they failed. It's really how the brain actually protects against those. So it's sequestering these toxic species in there. And our main advantage is that we protect the good guy, right? So really the basic biology, we described it in our last paper, where we kind of defined what we call the single toxin theory of neurodegeneration. It really unifies, right, kind of all these neurodegenerative disorders, because we start with very high metabolism of the brain. It's about 20% of our energy we burn in the brain. It's only 2% of our body. And we produce enormous amount of byproducts, about 1.5 pounds, about actual weight of the brain. When we get older and the brain is not able to, clear all those byproducts, some of these proteins that have a very important normal role, like amyloid, get stuck. They have a tendency to change shape and again start forming these toxic aggregates. And our molecule, as a small molecule, is really protecting this good guy. Now, the monomeric amyloid, which is again very important discovery that the Alzheon team has made, is really forming, kind of the first defense in the brain. It's one of the most important systems that the brain has to protect against injury, infection, stress. And it's preserved evolutionary for hundreds of millions of years, in the brains of lizards when they were getting out of the primordial ocean. And really, you want to actually have the system in the brain. So the problem is that some of the approaches that try to stop the production, like beta secretases, gamma secretases, which we in fact worked with this team on a previous company. We were the first to introduce beta secretases to the clinic. I did a $1 billion deal with Stellas. But you really want to have an approach that protects this good guy and prevents the formation of these toxins. And that's really what we're doing. Now, to me, the biggest surprise has been that we see the effects we do in the studies that we have completed in the patients who already have 20 years of actually accumulating the damage, right? So, so even if you intervene in the terminal state, it still appears that this mechanism is so profoundly impactful that if you can stop it, you will help patients. You'll see the effects on the biomarkers, but you potentially can help the patients. Now, again, our product is finishing the phase 3. That's the cardinal experiment that we're all waiting for, and that really going to be the ultimate validation for the approach and for this molecule. But again, the advantage we have that unlike the antibodies, we're trying to disable the toxin or clear the toxin and so forth, we're preventing the formation, and we can do it at a very early stage. And by the way, the other thing, there was another discovery from the Alzheon team. We have in the brain a system that protects amyloid, the monomer, the good guy, which you would expect, because if it's so important for brain function, there has to be something. And probably the one system that protects it is the one that we're boosting with our treatment. So that's the reason why our drug has such a good safety performance. We have over 3,000 patient data set followed for up to 3 years, and the drug, really, other than some of the GI side effects, has had a good favorable safety profile, in particular not having the brain edema and hemorrhage increase and so forth. So again, our main advantage is the interacting at a very early step, protecting the system that normally you need in the brain, but not allowing it to go to and supporting the clearance of the amyloid from the brain.